Okay, good. So here's my contact information. On here, I'm in the new life sciences building. My office is LSB 311. For those of you who used to visit me in my old Snyder office, this is a big upgrade. So I actually hope that some of you come and visit me so I can show off the new building, because it is a wonderful building. Some of you have probably already had lab classes in it last semester. So office hours. My office hours are by appointment. They're either through Zoom. I'm happy to meet with you one-on-one over Zoom, or if you want to come in and sit down with me and talk, I welcome that as well. But either way, all you need to do is send me an email and we can find a time when we can connect. Okay, so I leave that up to you. I don't have standing office hours right now because I kind of found out that standing office hours with Zoom is kind of a waste of time, because I sit here for an hour with Zoom open and nobody shows up, right? So it's better to just do things by appointment. Okay, but that at the same time, I welcome you to reach out. All right. So to start off the semester, what is cancer? So I think all of you, when you hear the word cancer, have certain types of words that come to mind, right? So maybe take a moment right now and let's share some of those words. We can do it using the chat. All right. So if you want to just go ahead, each of you, and open up your Zoom chat and just come up with a few words that immediately come to mind when you hear somebody say the word cancer, right? And we can take a look at those. Okay. So I see some of you are typing in there. Good. So we have words like, for example, unregulated cell division, right? Words like prognosis, tumors, yes, proliferation of cells, malignancies, astrology. Okay, that's an interesting one, right? Because absolutely. Right? Uncontrolled cell division, uncontrolled cell growth, overgrowth of cells, mutations, right? That's a very important one, a very good one. Here are several words: bad, right? Or tumors, right? Genetic malfunctions. Somebody writes, just kidding about the astrology comment. No, don't be kidding. That's a good one, right? Because we'll actually talk about the connection there and where that comes from. All right, good. So those are all good words, right? And I totally get it, because when you hear the word cancer, I think you immediately think of negative things like tumors or deleterious mutations, right? Harm, genetic malfunctions. Let's go back to the share here. Okay, so all of those words, I think, are part of the sort of common feeling about what cancer is. Right? Many of those words are words that the general public, if you were to interview somebody on the street, they would say those words as well. I think some of you, because you are life science students and you've taken courses, you get a little bit more specific, right? You talk about things like genetic components of cancer, uncontrolled cell proliferation. You use terms like that are more scientifically specific. During the course of the semester, I think we will learn about all of these things, right? And hopefully, through our discussions, you will not only have a much broader understanding of what cancer is, right, but you'll also have a broader understanding of the context of cancer. Right? Not only what is cancer, right, but why is cancer even a thing? Right? And why is cancer actually a very important part of concepts like evolution? So to start off with today, today's lecture is kind of a softball lecture. There's not a lot of really tough stuff, but it's a good introduction. So an understanding of cancer begins with appreciating the relationship of the words genotype to phenotype. Okay. I think all of you already have a sense of genotype versus phenotype, but let's get on the same page here. Back when Gregor Mendel was doing his experiments with pea plants, and he was the sort of the forefather of modern genetics, what we call genetics now. All right, back then there was no such thing as genetics. He was the first one to propose that the information that is passed on from one generation to the next generation, from parent to offspring, is somehow transmitted as individual packets or particles of information. Right? And back then, he was talking about certain types of traits that were pretty readily visible to him in his little pea garden. Right? So things like different phenotypic traits of round seeds versus wrinkly seeds, the color of the seeds, the color of flowers, things like where on the pea plant the flower appears, different types of attributes having to do with the shape of the seed pods, right? Either inflated or pinched, green, yellow, or even things like the height of the plant. Right? So all of these things, I think you are aware, are encoded for by genes. And the specific genes, those specific sets of information, are the genotype of the organism, of the pea plant. Right? How those sets of information end up being expressed in the real world so that people like Gregor Mendel or ourselves can see these different attributes or traits like colors, shapes, height, things like that, right? That is phenotype. And so undoubtedly, there is that indelible connection between phenotype and genotype. Right? And that will be at much of the core of our discussion throughout the semester. So let's start thinking about our genome, right? Because this course is mainly focused on human cancer, so it's only appropriate that we start off right away, starting to think about the human genome and our phenotype. When you think about something like the entire size of the genome of an organism, we now know that the size of the genome in terms of number of DNA base pairs doesn't necessarily correspond to how complex the phenotypes are. Right? So something, for example, when we talk about these terms, genotype, phenotype, and allele, right, this is where these words come from. So genotype comes from Greek, genos, meaning origin, right? Like Genesis, right? I think that's pretty obvious. Phenotype, maybe you don't really realize, it comes from the Greek phino, which means shining or the appearance of something. That's why we can think about it as the genetic code as being the origin, the genotype that gives rise to the outward appearance of an organism, right? The phenotype. But of course, I think all of you realize that phenotype is not just about what you see, it's also about the many myriad functions that happen inside of your cells, right? That's also phenotype. And then lastly, alleles. Allele comes from Greek meaning allos, which just simply means that another of the same kind. Right? So different alleles or different variations in genes give rise to the creation of the same protein, or that's what's meant to be the same protein, with maybe slight variations that make one version of the protein shaped one way, another version, another allele, causing a protein that gets encoded from that to be slightly different, but yet they're still meant to be the same protein. Right? So when we think about genes and how they're arranged on our genome, remember that genes are specific regions of DNA containing transcribed information. If we think about something like a shopping receipt, right, that you get at the cash register, you look at a receipt and it's just a whole bunch of code, right? In this case, the letters of the alphabet, the 10 digits from 0 to 9, various types of punctuation, like hash marks or commas or exclamation points or asterisks, right? If you couldn't read, if you were completely illiterate and you looked at this, it would just look like this nonsensical long list of symbols, right? But because we can read, we can sort of almost automatically, without thinking about it, tell that there is information in there. Right? And importantly, that it's not just letters and things like that that represent specific items on the receipt, but different types of coded symbols that let you know where one set of information starts and another set of information ends. And so from that, if you buy, let's say, a full basket of a dozen different items from the market, right, you see them listed on the receipt. And if you know how to interpret it, right, that information is contained there. The amazing thing about our genome, of course, is that it is analogous. It is a big, long list of instructions with different types of symbolic kind of codes that represent where things start and end. But the amazing thing about it is that rather than relying upon the full alphabet and numbers and a whole bunch of different symbols, your genome does this just by different combinations of A's and T's and C's and G's. And that, I think, is truly astounding, right? I never cease to be amazed when you think about it that way, that the complexity of who you are and the complexity of all living things on this planet just comes down to how the arrangement of A's, T's, C's, and G's are strung together in these molecules of DNA. So as a quick refresher, when a gene is transcribed, here is a region of DNA on a chromosome. And this particular region contains a coding sequence that is read by a protein complex, in this case, RNA polymerase II, that, remember, transcribes that message into an RNA strand, right? So it's sort of like a good photocopy of the original DNA. And the reason we evolved to do that is because now you can take that photocopy and you can edit it. You can edit it in different ways to remove certain parts of it and maybe combine different parts together. So introns and exons get spliced, and that way you're able to make a finished messenger RNA that then is exported out of the nucleus into the cytoplasm, where ribosomes can then handle the work of translating that, right? Taking that genetic code and actually interpreting it, translating it into a string of amino acids that forms a peptide or polypeptide chain that then folds together and forms your protein. Okay, so hopefully that's a review for all of you. Right. One thing that oftentimes we forget is that most of the genome is actually non-coding. That means it doesn't necessarily represent some message that's going to be transcribed and translated. If we were to think of this cartoon here as just one section of one of your chromosomes, so one continuous strand of DNA here, what you would find is that the red regions here are the actual genes, right? And there's maybe only a few of them in this particular stretch. In this case, there are only 5 genes that are encoded for here. The rest of it is just what we can think of as this yellow colored region. Junk, right? In the past, I think 20 years ago or so, that's what scientists thought. They just thought it's junk DNA. It's somehow remnants of evolutionary editing that left us with stuff that we don't really need, because the stuff that's really important are these red regions, the genes, right? But now we know that's not true. Even though only 3% of the entire human genome actually codes for proteins, so those would be the red regions in here, we now know that the other 97 or so percent of non-coding DNA is very important. A lot of that is truly junk because there are many repetitive regions that are in fact artifacts of different evolutionary sort of moments in time. Many of them are repetitive regions that are a result of things like viruses becoming incorporated into our genome over evolutionary history, but they are no longer functional as viral encoding regions, but they're just kind of left there as sort of like baggage, right? But much of this non-coding region is in fact important as regulatory regions, regions such as sites of enhancers or repressors, where different protein transcription factors are attracted there, build up, and then the effect of those proteins are then physically brought to something like the promoter of a gene thereby controlling the RNA polymerase activity to transcribe that gene. Okay, and we will study that in more depth as the semester proceeds. So the reason I wanted to introduce this right now is because a few of you mentioned this concept of mutations, right? So mutations are, of course, alterations in the genetic code. So these can either be an individual DNA base pair that gets altered. For example, there should be an A there, and instead there's now a C there due to a mutation. Or it could be larger chunks of the DNA actually get accidentally deleted or even swapped around between different chromosomes so that different information is no longer where it should be in the genome. Okay. On the left, you would have an example of what we tend to think of as a selected mutation. In other words, a mutation that actually has an impact somehow on whether or not the resulting mutated genome is considered desirable evolutionarily or undesirable. So in this case, let's say we have a coding sequence and you have a mutation that happens right in the middle of the coding sequence, right? So as a result, this version of the gene has now been altered in a slight way. So we say that it is now technically a different allele, right? It's the same gene that's meant to code for the same original protein, right? But now it's an altered version of it. So we say functionally it's a different allele, and maybe it's defective now, right? So maybe this single DNA change results in a change in amino acids at that particular position in the polypeptide chain. So as a result, when the finished protein tries to fold up into its correct three-dimensional shape, you know, maybe there used to be something like a glycine at that position, so it wouldn't really have a big impact. But now there's something like a phenylalanine or something like that, right? So now as a result, the protein doesn't fold up properly because there is a bulky side chain there. Or it could be a premature stop mutation that causes the resulting protein to not be translated beyond that part. So you only get a part of the protein made, right? And as a result, that protein doesn't work. So let's say that results in a defective phenotype, then to some extent, it's going to have a negative impact on the organism as a whole being able to sort of carry out its normal functions, and it would have a negative impact potentially on that organism being able to reproduce or reach a point where it can reproduce and pass those genes on. So such mutations then might then be discarded from the gene pool. Right? The opposite, of course, is this doesn't have to be bad, even though the example they give is defective, right? It could be that somehow this mutation actually enhances the function of a protein in the context of, let's say, some big cellular pathway that this protein functions, in which case it may end up resulting in desirable changes to the phenotype that enhance the ability of that particular individual to reproduce, right? Or be considered an attribute that actually makes it more attractive for a mate to reproduce, right? In which case those mutations would be selected for. But consider also that mutations that happen in the yellow, more junk region of the DNA, right? If there's no big impact on things like the alleles or the versions of proteins that are made, then maybe these neutral mutations, right? Maybe they don't really matter. And because it doesn't have an impact, positive or negative, on the ability of the organism to be healthy and reproduce, these neutral mutations maybe just get carried on to the next generation and the next generation without having a big impact, right? So many of these mutations are what we now call polymorphisms, right? That's why you can actually have different profiles of these polymorphic locations in the genome that if you're able to profile what assortment of different polymorphisms or variations that you have, right, you can use that to trace your ancestry, because these mutations had no real impact on your ancestors' ability to reproduce. They just got passed on as part of the genome. Okay. Now, it is possible, certainly, that these mutations are actually happening in the middle of one of these regulatory regions, like an enhancer or something like a repressor, right? In which case, the effects on the expression of, let's say, the target gene that that enhancer is meant to control, right, might be subtly altered. So it'd be very subtle. It wouldn't be a defective protein. It might be just different changes in the level of protein that are made, right? And those could be beneficial or detrimental as well. Right? So that's why there's a lot of work now to try to understand whether some polymorphisms are actually associated subtly with different types of diseases. Not because they result in a defective protein, which would be more obvious, but because over the many, let's say, decades of a person's life, the influence of having a particular polymorphism on the levels of protein that are expressed by their cells over many years of life might actually shift them to be more at risk of developing certain types of diseases like cancer, or might actually shift them to be more protective against developing things like cancer, right? So it's kind of fascinating to think about, and we will explore that as we go on. So as a reminder, the chromatin in our typical G1 phase cell is very loosely organized in this illustration here, right? It looks like a bowl of noodles, right? So in fact, you know, okay, I'll just go with it, right? Because you know how I like to think about our chromosomes as being a bowl of Simon, right? And then if you look at these, the DNA is of course tightly organized as chromatin, not as just naked DNA. It's tightly packaged up with proteins such as the histone proteins that make nucleosomes. During mitosis, the DNA is much more compacted, and this is where we see the typical sort of X-shape form of chromosomes that I think the general public is usually more used to seeing. Right? But of course, this only happens during mitosis when you have the condensed chromosome, in this case, still paired up with its sister chromatid, the replicated version of it. And now this X shape is going to proceed to be pulled apart during anaphase. Just really quickly, I think for those of you who maybe don't go to grad school or med school, but end up wanting to do something else, I think what you should do is you should start your own line of geeky biology instant ramen, right? Like Top Ramen, but make the little dried ramen packs the shape of condensed chromosomes like this, right? I think there'd be a market for that. If I lose my faculty position, that's what I'm doing. Okay, all right. Anyhow, how physically long is the human genome, right? As a reminder, a single haploid copy of our genome, the one that you either got from mom or dad, if you were to string each of the chromosomes together end to end, it'd be about 6 feet long, right? That's pretty phenomenal if you think about it. I mean, that's like as tall as I am, but the point is you would not be able to see it because it's only one strand of DNA thick, right? So it's remarkable that all of that can be packed up, not just all of that, but a second copy of it, right? The one you got from mom, the one you got from dad, all packed up into the nucleus of an individual cell in your body. Right? Here you see a karyotype. A karyotype is, you've seen something like this before, is where researchers, let's say they want to try to examine an individual's genetic profile to see if there's any evidence of gross genetic abnormalities, for example, that might be part of explaining a disease phenotype. So they take cells from their body and they try to capture a few cells that are in mitosis so that the chromosomes are condensed, right? In this case, you can see there are what we expect, 46 pairs of chromosomes from the largest chromosome 1 all the way down to the pair of sex chromosomes. In this case, it's a male, so there's an X and a Y, right? And so you have the 22 pairs of autosomes. This type of visualization is done by FISH, which is a technique whereby different small little probes or pieces of DNA that are specially designed to be unique to a particular spot in the human genome are labeled with fluorescent molecules, so that if you then go ahead and stain a cell with these, right, presumably each of these little markers will only fit and bind to a particular spot in the entire genome. And so if you design it so that all the ones that bind to chromosome 1, for example, fluoresce yellow, right, then you end up being able to have a visual representation of chromosome 1. You can see there are some spots where it looks like there's like a little glitch, right? There's a little green here on this one. If you look at the green chromosome, where is it here? Maybe there's a little bit of brown or red on that one. And that's because some of these probes, maybe are a little bit too similar to different sequences that are on different chromosomes. And so you get those little glitches. But by and large, this is what a normal healthy cell karyotype looks like from your body. Right? Obviously, I'm talking, in this case, it's XY. So for the women in the class, you would have two X chromosomes, right? So if this is what a normal cell looks like, what does cancer look like? A normal karyotype looks like this from a diploid cell. Cancer looks something like this. Right? So these are abnormal chromosomes. In this particular case, it comes from a patient who has pancreatic cancer. So this is from a biopsy of a pancreatic tumor. And you can see a number of different things here. The most striking, of course, is that many of these chromosomes now look like candy canes, right? They have different regions that are clearly not just little glitches in the probes, not finding the right site. These are entire sections of wrong chromosomes that have been swapped with other chromosomes. And the other thing that hopefully is kind of obvious to you is that the ploidy is completely messed up. This is no longer diploid. It's certainly not haploid. It's the opposite. It's multiploid, right? What we call polyploid. So this particular cell now has 4 copies of chromosome 1, for example, right? All of this, whether you're talking about wrong regions of chromosomes swapping around, or just having abnormal numbers of a chromosome, right, go far beyond simply having a little point mutation that alters an allele of a gene, right? This is kind of extreme now. So think about what effects can this have on gene expression? The most obvious might be you might have overamplification of genes. Certain genes that are meant to be expressed in certain levels, right, now suddenly get blown out of proportion in terms of their levels. At the same time, you may lose expression of certain genes. And because these genes are now in the wrong place on the wrong chromosomes, just think the coding regions of these genes are now maybe inserted in places where the non-coding regulatory regions around them are the wrong regulatory sites, right? So that can also have a big impact on the expression levels of these genes. Okay, so again, these are all things that we'll talk about this semester, but I wanted to introduce this to you as sort of a very obvious, sort of hopefully kind of almost jarring way of taking a look at what mutations are all about. So to kind of get us all on the right sort of start here, I really think it's important to think about the relationship between genotype and phenotype, because it's very simple to think like, oh, depending on what genotype you have, that dictates what phenotype you have, right? But it's not true that genotype alone equals phenotype. Normally, in-person semester, I would start off this class by passing around these 2 bags of Arari crackers. I always say it's nice to have kind of break the ice by having everybody get off to a nice start and have a little snack, right? Of course, we're online. We can't do it, but I still have them here, right? And so let's pretend, though, that I pass these 2 bags around and everybody takes one of each, and you have it on your desk and you're looking at it. Right, so these are very typical Arari crackers that I think all of you are familiar with. This one here shaped like little cherry blossoms, the sakura. This one is shaped more like these little sticks, right? So the question is, these are obviously very different crackers. They have different phenotypes. There's no doubt about it. Let's imagine that these are normal, healthy cells in your body, right? Your body's made of 250 plus different types of cells. Let's pretend that these are 2 examples of them, right? And you might say, well, clearly, right, these are very different. The instructions must be very different for making these. But how different is it really, this idea of genotype being linked to phenotype, right? You cannot really see the labels on these too well here, but let's pretend we pass these around like I normally do, and then it ends up with a couple of you, and I ask a couple of you to kind of read off the ingredients. So let's say maybe Darcy, if you want to unmute yourself, and Darcy, I sent her a picture of the label for the these packets here. Do you want to read the ingredients of the sakura crackers?
Okay. So the sakura arare has glutinous rice, soy sauce, which consists of water, soybean, salt, and wheat, sugar, fish extract, which is bonito extract, and mirin, consisting of rice, alcohol, enzyme, and water. Water, salt, monosodium glutamate, glucose, hydrolyzed vegetable protein, bonito flour, sugar, xanthan gum, disodium inosonate, and disodium guanylate, caramel color, and modified tapioca starch and seaweed.
Okay, good, thanks. So aside from the fact that there are some really chemical sounding things like that, you might think I'm going to be talking about them as being carcinogens. We'll save that for a different lecture. What I want to do is really think about the main ingredients, right? So Alyssa got the other pack here, the little mini nori ones. Do you want to go ahead, Alyssa, and read the ingredients to that one?
Yeah, so in the mini nori maki, there's glutinous rice, soy sauce, which has water, soybean, wheat, salt, sugar, tapioca starch, caramel color, sesame, seaweed, monosodium glutamate, bonito fish powder, salt, ribotide, and chili.
Okay, wonderful. Thank you. Both Darcy and Alyssa have been TIs for me in the past, so I know that they're very good at chiming in and participating in the discussion here. So the point I want to make, okay, is aside from the very chemical sounding little preservatives, the main ingredients of both of these on the label are rice, soy sauce, seaweed, sugar, there's modified starch, there's fish oil, etc. Right? So by and large, both of these crackers have the same ingredients, but yet the phenotypes are very different. And that's the point that I want to make here, right? Because where does this difference come from, right? If in fact they have the same ingredients. So think about things like this. There are many factors that affect what kind of phenotype you get from more or less the same genotype. You can think about, in the case of the crackers, maybe the supplier of the ingredients that each of these different manufacturers, right? One is the Hawaii Tradition one, the other is the Enjoy one, right? I actually like all the dried seafood stuff that the Enjoy ones make. But anyhow, the supplier of the ingredients, right? So that might be analogous to, in the cells of the human body, the genetic background, right? Where you're actually getting these ingredients from, right? Then the amounts of the ingredients. In one case, you have a little bit of nori on the left, right, with those sakura crackers. On the right, you actually have much more of the nori. So that has to do with expression levels of those ingredients, or the expression levels of the different genes. Interactions of the ingredients. For example, I imagine the nori in the crackers on the right still are kind of crisp, right? Whereas the nori flakes on the one on the left are probably kind of soggy because they're all just part of the dough, right? And even the packaging. One package, this one here, right? Once you open it up, you're going to have to put a little clip on it to keep it shut, whereas this package here, right, has a little zip thing, right? So all of these correspond to things like the genetic background, the gene expression levels of the different cells in your body, the interactions between different genes, right? How is it that having way high levels of one protein might affect a different gene's expression level, and also environmental exposures. In the case of these bags, right, it may mean the difference between keeping one set of crackers crunchy and the other set of crackers getting stale too quickly. Right? Likewise, in our bodies, environmental exposures can have a big impact on how the cells actually express phenotype, right, and what you start seeing. So the point is this: if those two crackers, right, are the normal phenotypes of, let's say, two example cells in our body, then what is disease? So this third one here, right, you say, ooh, normally I'd be excited and think that looks yummy, but now that you're calling it disease, I'm grossed out by it, right? No, but the point is, clearly, this is no longer this one or this one. It is something else, right? The question is, where did this something else come from? If we were to say that this thing came about starting from one of these, which one did it actually come from, right? Is it, well, you say, well, it has a lot of nori, so it probably came from the one on the right. Or you say no, because look at the filling. The filling is much more like the one on the left. This is a very important concept when we study the etiology of cancer. Because what we're saying is, if you're going to try to understand the biology of a particular type of cancer and cancer cell, a big part of the puzzle is knowing what the original normal cell type of origin is. Okay? Because knowing the normal cell type of origin gives you a lot of insight into the likely cellular pathways that are in the cell that are responsible. Right? And if you can identify or have a hunch as to what is going wrong inside of the cell, then you have a much better understanding of potentially how to treat that particular type of cancer by targeting things like gene expression pathways that are more relevant for the Sakura type of cracker versus the rod-shaped type of cracker. And that can make a profound difference on the success of a patient's treatment when time is of the essence and they present with disease in the clinic and you don't have weeks or months to just guess. Okay, so we'll go ahead and end our intro lecture there today. On Wednesday, what we will do is we'll take a look at how different types of cancers are actually categorized formally by oncologists to give us a better picture of how complicated this whole question really is about normal origins of disease leading to a multitude of different types of cancer phenotypes and cancer diseases. Okay, so let me go ahead and stop